prevention medicine as tailoring of medical treatments to the individual characteristics of each patient. And then if we want to expand on that broad definition more broadly, we can say that prevention medicine is when you give treatment that target the needs of individual patients on the basis of their biological makeup in terms of the genetics, which is something functional where they have been, what they have been exposed to, the phenotype characteristics, the psychosocial characteristics. Every feature that distinguishes one patient from the other, and tells also clinical presentation. So there are some other technologies that are used interchangeably in prevention medicine. These are personalized and individualized medicine. Now, the prevention medicine concept really became popular or popularized in 2015 when the then President Barack Obama of the United States launched the Prevention Medicine Initiative. Then director of the NIH Institute, National Institute of Health, Dr. Francis Collins, wrote this elegant commentary in the New England Journal of Medicine to again further popularize the concept in the US. But now, as my director said, the concept of prevention medicine is changing the way we think about healthcare. Please, can I confirm that you can hear me clearly? And that you're following? Dr. Bakary.
Hello, we can hear you, but it's breaking.
We can hear you. We can hear you, sir.
We can hear you. You only that you are breaking. Go on.
We can hear you, sir.
Hello, Dr. Akinyemi. Is changing the way we think about healthcare. If you look at this, it's been. Hello?
Yes, we can hear you, Dr. Akinyemi, but if you can maybe
just go on.
Maybe it will be better. Just move around. Maybe your network will be better. Can you hear us? Professor Akinyemi? Professor Akinyemi, we can.
I'm no longer here.
We are not hearing. You are muted. Professor Akinyemi, you are muted. Unmute.
All right.
Hear you. We can hear you. Okay.
Right. Okay.
We can hear you.
Okay.
Go on.
Right. Thank you. So now if you look at this slide, it's echoed what my director started with, that prevention medicine is really changing the way we think. In the past, medicine was more of an intuition, endeavor. You listen to the patient complain of signs and symptoms, and based on your own knowledge base, you make a diagnosis and you provide treatment. But these days, we are talking about evidence-based medicine. We derive evidence based on clinical trials, randomized clinical trials, or systematic review or meta-analysis, talking about level 3, level 4 evidence, and we use that to develop treatment guidelines that guides how we treat patients. But prevention medicine is creeping in very quickly, whereby medicine decision will be made not just by our intuition or clinical trials alone, but a lot of algorithms that are based on technology and and another related infrastructure. So now precision medicine also, as I said earlier on, there are some other synonyms like stratified medicine, personalized medicine, individualized medicine. Although there are subtle differences in the definitions, but I won't go into the depth of that because of time. But they're all like twin or triplets of the concept of precision medicine. Now, so this is a very simple reductionistic, explanation of what of the concept of precision medicine. If you look at this panel, you can see there are 12 individuals on the left of the panel, and then you find these individuals in 3 different colors: blue, green, and orange. They all mixed together. So this can be likened to, let's say, a group of patients who have a cancer. Right. So in the current framework, one can fit all. You give all of them the same combination of chemotherapy for their cancer. But as you know, the outcomes will be different. Some will survive, some will die, and some will even recover from that condition. So there are usually different outcomes. But with the input of precision medicine-based technologies, including looking at their DNA sequence, looking at their epigenome, their metabolomics, their proteomics, including their exposome, the environment they are exposed to, including their lifestyle, their social network, all these are different other factors that you consider. So when you input all these variables, then you're able to stratify them into 3 different groups that are now homogeneous. And the treatment for the different groups differ. They're more precise. And of course, they give better outcomes and at the end of the day. So this slide again is just to illustrate what I've said in a few words using tutorial format. We had the one-size-fits-all medicine. Now you can stratify based on demographics, subtyping, histology, typing of the disease, and then biomarkers. Biomarkers are becoming very, very important to virtually every disease. And then when you go to personalization, then you take on more variables that are individualized so that you're able to actually deliver care in a personalized framework. Now, this slide is to show again, to echo the importance of data science to precision medicine. It's all about data. Generate data from your interaction with the patient, from the clinical records, from the electronic health records, from the case notes. Then when you take blood sample, get blood, fractionate the blood into extra DNA, store your plasma serum, rest concentrates. And from these stored products, you run different types of high throughput technology, next-gen sequencing, mass spectrometry, multiplex biomarker analysis, ELISA, different. And at the end of it all, what do you have as output? You have data, huge data. Data, that's what you have. And then, but you now need the framework, you need the expertise, the technology, the competence to now analyze these data sets, using different bioinformatic algorithms, and we even do what you call integrative multimeric analysis, and for this, so this is what data science is all about. And you agree with me that this also throws up the other ethical, legal issues, anthropological issues, political issues about data science. Who owns the data? If you've taken data from the patient, generate data in the library, does the patient still don't have the data? Or does the data belong to the placement investigator or to the institution or to the hospital or to the country? Then how can you share this data, across country, across nations? So the LC issues also come on board. And that's why data science is actually an important component. I just provide some further details about what I've described. With all the huge data set you have, you can actually phenotype individuals and then run integrative analysis and then generate clusters of patients, categorize them, and then be able to have these potential medicine outcomes or products that range from discovery of new drugs, diagnostics, therapeutics, prognostics, and of course, drug repurposing has also come on board as one of the outcomes or good outcomes of precision medicine. So, in summary, precision medicine wants to ensure that the patient gets the right treatment at the right dose, at the right time, with minimum consequences and maximum efficiency. That's the ultimate goal of precision medicine. And of course, it is also possible to operationalize the concept. Instead, in a population context. When you do this in population context, then we say precision public health. So you can apply the principles systematically to develop cost-effective target interventions to communities. We all know that this is what implementation science is all about. You take an intervention that is generated and that has worked somewhere, and you domesticate it in a different context to also achieve the same result. So it applies to persons, families, and of course, all communities, even nations. Now, there are certain health conditions, illnesses, for which the principle of precision medicine is already in application. And this cuts across different specialties, from cancers to blood disorder, infectious diseases, cardiovascular diseases, skin disorders, of eye disorders. This slide just shows example of those diseases. The biomarker that has been used for the stratification of subtyping, and of course, the interventions, some of which are targeted therapy, some are biologics, some are immune modulators, and some are actually new drugs, and some are repurposed drugs. So precision medicine is already alive, and it has come to stay. Now let's quickly proceed by moving on to biobanking. Now, biobanking is simply talking about biorepository. We do know that a biorepository is a material repository where we collect, store, and specimen in order to picture. Okay, can you please, if you're not speaking, please, can you please mute? Thank you. So, biorepositories can contain or manage specimens from animals, humans, and many other living organisms. Now, we tend to use the terminology biobank when we are talking about repository of human biological materials. You store those materials, including the policies and procedures that protect them. So in other words, biobank, tends to we use that term when we talk about human biological specimens. And biobanks can be based on the type of tissue being stored, whether it is tumor cell, blood fraction cells, DNA, or the purpose, research purpose, forensic, therapeutic, transplantation. But who owns it? The biobank, is it owned by College of Medicine or NIMA or a public-private partnership? By the time I mention some of the emerging biobanks in Western Council, you will appreciate the ownership framework better. And of course, it can also be based on the size. Is it a bank that serves a department, an institution, a state, a region, even a country? So it depends on the size. Now, permit me to talk about neurobiobanks. So now, I'm a neuroscientist. So we talk about neurobiobank. So basically, a repository of central nervous system tissue, associated blood fractions, CSF, including neuroimages. And biobanks can operate singly or they can operate as a network. And we have some of these examples in the US and the UK. But biobanking has come a long way. we now have an organization known as ISBA, International Society for Biological and Environmental Repositories, that now serves as a gatekeeper, or as it provides guidance, for biobanking activities globally. And I must tell you that this ISBA is gaining popularity globally all over the world day by day. And one of the things ISBA does is to provide best practices, guidelines for best practices in biobanking science, ranging from how to plan your biobank, your storage facilities, quality management, safety training, records, scores, biological material transfer, specimen collection, processing, retrievals, standard operating procedures, and of course, packaging and shipping, and also not leaving out, ethical, legal, and social issues. So there are guidelines that ISBA has developed, and every existing or new biobank is meant to work with those criteria. And even recently that we began to interact with the National Health, the IAMRAT, that's the Research X Committee of Nigeria. I realized that when we started interacting with them, that many of the principles about biobanking is also guided by the ISBA best practices. Now, we've talked about precision medicine and we've talked about biobanking. Now, let's now try to situate precision medicine within the context of African genomic diversity. Now, the African genome is the most diverse of all ancestral genomes. In other words, if you take a European, an Asian, or any other ancestry, the genome of the African is the most diverse. And this slide is a reductionistic approach to just explain this. You see that if you compare the genome of the non-African to the African, you can see that for the African, you can see the approach to that of the non-African. And then we also say that the liquid disequilibrium is low, it's lowest compared to other richer populations. And people who have done elegant work like teach course work have documented the variation in the African genome. But the interesting thing is that the genome is not only diverse in comparison to non-Africans, but even within Africa, there is a lot of diversity. In fact, our genome is as diverse as our language, our geography, and our culture. That's what this map shows. And this slide shows these are all Africans from different cultures, different ethnic backgrounds.
This one is to go into your brain.
And the in fact, Stratishkov, very renowned anthropologist and geneticists, did say that the genetic difference between an Englishman and is smaller than that of difference between two Africans. That shows the level of diversity of the African genome. The question is, why is our genome so diverse? Well, I won't have all the time to go into deep explanations, but this study is just meant to provide some understanding. We're diverse because of diversity of our geographical location. The African environment is so diverse, cut across the equator from the north to the south, different environment, language differences, different patterns of subsistence, dietary differences, multiple migrations, and then survival related to exposure to infectious diseases. So there has been a lot of intra-African migration, the out of Africa migration, then back to Africa migration. And we even know, even from some of the folklore stories that we hear in the environment, we hear of maybe in Nigeria, the Egbas left Ife, went to present location. The Benins left Ife and went to the present location. So we know because in the tribal wars, people looking for a place of refuge, people move. And when people move to different places, they intermingle with other genes. So there's a lot of genetic admixture. And of course, there's also survival issues, survival of the fittest. So that these are all factors that are responsible for the diversity of the African Union. And of course, it also believed that man, was first, the Homo sapiens was first found on the African continent. And so as man migrated out of Africa, then the genome became more homogeneous. And this happened hundreds of thousands of years after the original man was located in Africa. So, but what are the implications of this genome diversity? If you look at this panel, on this left, you could see that this is based on the 1000 genome project. You could see that when you run what you call principal component analysis, it's a way of disentangling population structure. You could see that the Europeans and the East Asians, they kind of segregate together to the right, and then the Africans segregate to the left. Now, but if you can see that even the Africans, the Yoruba, the Khoisan, people from different, we could see the distance between the different groups within Africa. So that is a reflection of how diverse we are as Africans. Then if you look at the second panel, the liquid distribution is lowest compared to that of Asians and Europeans. And then if you come to the panel below, if you look at the heterozygosity ratio, the variance site reference genome, you can see that of the Africans is a lot higher compared to that of the Europeans or that of the Asians. So these are all strong risks that demonstrate the huge diversity of the African genome. Then what is the implication of this diversity? One of the things we do with genetics is risk prediction. We can predict the risk of disease by looking at the totality of the genetic contributions to a particular disease. So when polygenic risk scores are developed in Europeans, if I apply them to Africans, you can see that it is least accurate. Both the panel to the top right and top bottom shows that polygenic risk scores that are derived from European populations are least accurate when you apply them to African populations. And this is the reason why we need to develop our own Afrocentric, our African-specific polygenic risk scores for diverse conditions that we see on the continent. Now, the other implication of the diversity of the African genome is that when you include the African genome in a global genome analysis, the African genome helps with the causal variants of diseases and also helps with fine mapping so that you can actually more carefully localize the genetic variants that are responsible for disease. And that's the reason why all over the world, there's a huge hunt for African genomic data. That's why everybody wants to get DNA or from Africa, because when you introduce African genome into the equation, it helps. It makes it the prediction, determination or causation of other diseases, more accurate. And that's why this Nature paper published in 2020 said that greater availability of African genome will improve global understanding of genomic variations and complex traits associations in all populations. So it will be a blessing to even other populations when they take part in global genomic endeavors. And that's also the reason why there's a lot of push for diversity, equity, and inclusion in the genome world. Now, let's come closer home. How much do we know about precision medicine among us as Africans? I found this study interesting. It was done among a group of South African clinicians. And their perception of precision medicine regarding cardiovascular disease is that precision medicine will make available an African CVD risk tool. That's the risk prediction tool, like the polygenic risk score. And the precision medicine will also enable us to adapt prevention and treatment approaches to a more African friendly environment. And of course, it will also enhance risk estimation and to an incentive to adopt lifestyles that are suitable for particular individuals. And of course, information for patients. However, there are barriers. There are implementation barriers to domesticating patient medicine in Africa. These range from issues about cost, about human resources, about infrastructure, and of course, cultural concerns. Now, this slide is also taken from that Nature Review publication by Pereira et al., talking about the challenges, enablers, and benefits of patient medicine in Africa. You can see that the challenges again range from high disease burden, our genetic heterogeneity, inadequate infrastructure.
Shiny drinks, too. I better come.
Can you please mute, sir? Death of skills, inadequate data, and all, whereas the enablers will be political governance, data sharing, longitudinal cohorts, ethical oversight, and of course, digital technology. All this will enhance patient medicine on the African continent. And of course, this will also work through the principles of implementation science, research, infrastructure, cultural data knowledge, and doing feasibility assessment. And all this will deliver impact both at population and individual level. Now, this data resource, known as the shared data resource, is a all right, thank you, sir. Thank you. Now, this is a resource of about 55,000 individuals who were, this data is a huge data set put together within the East Africa Constitution under the leadership of Osman Awolabi when he was chair of that working group. And we can take advantage of this. It's a big data, and data set like this can enable us to catalyze the achievement of this in Africa. Now, I want to come closer home. Can you please mute, sir, if you're not, can you please mute if you're not speaking, please? Now, precision medicine, permit to lay the two focus on precision medicine and brain disorders in Africa, especially looking at stroke and dementia, which are our key areas of research in this environment. In Ibadan in particular, there are areas of strength. Neurogenomics is also coming of age, especially relating to stroke and dementia, as we do in Ibadan. For dementias, it is well known that there are racial differences in the use of Alzheimer's disease. For instance, we do know that in the US, African Americans and Hispanics are more likely to develop Alzheimer's disease compared to people of European ancestry. But what was found in the Indianapolis study was different. There was lower incidence and prevalence of AD compared to African Americans in the US. So this again highlights the interplay of genes and geography, genes and environmental factors, including lifestyles, playing out. And we don't know, of course, that the high prevalence of cardiovascular risk factors would accounted for the higher burden in the African Americans. This was a neuropath study done in Brazil. Brazil is a multiracial population where you have people of African ancestry, people of European, and people of Hispanic ancestry all together, all still outcomes of the slave trade of the 2-3 centuries ago. So what is shown in this study was that among people of African ancestry, the African ancestry protects against Alzheimer's pathology. So that the odds ratio for Alzheimer's pathology, neurologic plaque and neurologic tangles is less than, meaning that people of African ancestry, they are less likely to lay down amyloid. But if you look at the odds ratio of small vessel disease, you can see it's higher than 1. So meaning that vascular factors may be the primary driver, or may play a stronger role in this, in that population. And Our understanding of this has also been be further expounded by this recent publication in which it was shown that, like, if you look at the panel above, it's talking about the contribution of the apolipoprotein E4 allele. How does it contribute to the risk of Alzheimer's disease? You can see that the contribution of that allele is highest among the Japanese, followed by Europeans, then Americans, African history, and then lowest among indigenous Africans. The question is, what is it that is attenuating the Alzheimer's disease causing effect of ApoE in this population? It is because of a novel African ancestry specific locus that is protective, that was recently discovered, in this study. And this study, the study included some data from the Biobanking Indianapolis project. It was meta-analyzed with data from Latin America and North America. So again, this shows the ancestry differences in risk of disease, and of course also highlighting the importance of precision medicine to be developed for different racial populations. And I must tell you that now we are exploring some of these findings further in the current largest study of dementia of the continent, known as the REDDSP study. We are recruiting 5,000 people from 9 African countries, 2,500 AD and 2,500 controls. It is hoped that this huge sample size will enable us to answer more questions and, of course, to find more discoveries to understand why our dementia biology is different from that of the Caucasians. Now, vascular factors are also very important. We know that whether you talk about Alzheimer's disease or vascular dementia, vascular factors are very, very important. They drive the production of amyloid in the brain, and we have done previous pathological studies to confirm that. There are also shared genetic contributions to ischemic stroke and Alzheimer's disease. In other words, both ischemic stroke and Alzheimer's disease have some shared genetic variants. So, in other words, the vessels are also important. The carry blood to the brain that's important as the brain tissue itself in terms of dementia risk. And this brings me now into our journey into the brain vessels itself. As you know, that the science study, legally led by Prof. Olabi and Prof. Agili, is the largest study of stroke on the continent with elegant outputs in the last decade. We have summarized some of what we know about stroke on the continent recently, including some of the recent inputs, contributions from the SIREN project. Now, there are some peculiarities regarding stroke in African populations. We know that among Africans, age of onset is relatively younger, about 60 years, almost 2 decades younger than what we find in Caucasians. We see more hemorrhagic stroke, more bleeds, and we have more small vessel disease, and we also have high genetic heritability. And we also have worse outcomes, some of which we believe not to be due to only social determinant of diseases. We think that genetics may also play a contribution. So we're eternally grateful to the East Africa Consortium funded by the NIH and the Wellcome Trust that enabled us to take a dive into exploring the architecture of stroke, genetic architecture of stroke in the Africans. Our effort was further supported by this team from the US, saying that why it is important to conduct stroke genetic studies in the African ancestry populations because of our genetic heritability, and of course for better resolution for file mapping and trans-ancestry meta-analysis. So this is the framework of the SIREN study. 3 major themes: the phenomics, trying to understand the phenotypic characteristics of stroke in Africans, genomics, and of course, the biostatistics and bioinformatics, all focusing on data science. And of course, we have stored materials in biobanks in the last decade, and they're still doing very, very well. We've conducted gene studies as well as our discovery GWAS. Some of the key findings of the SIREN study are shown in this slide. For the first time, we reported an association between the APOL1 gene variant, which is a variant that's normally associated with trypanosomiasis associated with, again, with chronic disease in people of African ancestry. We were the first to record an association between variants of this gene and small vessel disease, ischemic stroke. And of course, we also found an association between inflammatory genes like interleukin-1 and other related genes, showing that these genetic variants are very important in Africans. We've also conducted the GWAS, and our findings are also unique. Some of our findings, we found hits on chromosome 3 that are of genomic significance, and of 5. And then we found some very important associations on other chromosomes as well. But the important thing is that the associations that we found, the genetic cluster that have been associated with ischemic stroke in the siren cohort, providing they're suggesting regulatory rules for microRNAs, intergenic non-coding DNAs, and tonic non-coding RNAs. Some of these findings are very, very unique, and they've not been reported in other populations amongst whom strict genetic studies have been done. We made an attempt to replicate our findings in other populations, African Americans and European population, the COMPASS and MEGASOC study. But because we are different, we were unable to replicate our findings amongst them. Transferability was not achievable. And that's what is really saying, that this siren cohort and the megastroke, they differ in terms of allele frequency, and they show the fundamental differences by genomic makeup compared to Europeans, even African Americans. Not totally, there's some agreement, but not the perfect one. So, what are the functional implications of our siren GWAS findings? We found differential rules for microRNAs, intergenic non-coding DNAs. We also found some protective role for some of the loci. They are protective, almost similar to the protectiveness that was found in the AD ancestry-specific gene. And of course, some of these variants showed expression in brain, vascular, cardiac, and neuronal tissue. And of course, given these findings, we know that we're currently trying to explore the multiomics and of course develop an Afrocentric progenic risk score, just as I've explained earlier on. An attempt has been made through the phenomic data. We have we have been able to develop an Afrocentric stroke risk prediction just based on risk factors alone, hypertension, diabetes, but of course, with the GWAS being out now, we'll be able to fine-tune this and develop a progenic risk score in the nearest future. So I've tried to now use our own experience in neurogenomics, stroke and dementia in particular, to emphasize or to explain or domesticate how we can domesticate precision medicine in the context of African genomic diversity. Now, I'll quickly, as I'm rounding up, I'll quickly highlight some of the new frontiers in biobanking in the West African region. There are several biobank initiatives in the sub-region, some of which are home even to our College of Medicine. The Institute of Human Biology by the Repository is funded by the East Africa Consortium. It's in Abuja. And of course, we have the 54 gene, which is a genomic startup in Lagos. Then the MRC Gambia has had a repository for years. It's a strong research organization. Then following the Ebola outbreak, Lagos was actually able to start a biobank. It also became very useful during the COVID-19 outbreak. So the NCDC also has biobanks in Abuja and Lagos. Then within our College of Medicine, IAMRAT has hosted biobanks also for several decades. The Department of Virology, and of course, the last decade, Sarin biobanks have come on board. So also the MadCap project looking at, say, prostate. And of course, the Biobank project is also a fledgling biobank. And the College of Medicine is also developing a centralized biobank. This idea was initiated by CRIM originally. So just some quick lessons we learned from the Sarin Biobank. Again, I've shown us the framework earlier on, where we have published our experience with the Sarin Biobank in this publication, which I will encourage you to consult. The samples are collected from multiple centers, up to about 15 in Nigeria and Ghana. And we have a very, very rigorous and standard operating procedure. And we follow this flow chart in all our sites for sample collection, sample processing, sample storage, sample archiving, and even shipment to Ibadan, of course, to research partners when we needed to ship samples. So this standard operating procedure guides all our operation, and it's been very, very effective in the last decade. We also have a procedure that are solar inverter powered to ensure that the power supply is maintained 24/7. And of course, our DNA quality have been of the highest standard, and they have actually been validated by the higher in Abuja, to which we send about 6,000 DNA samples. Currently, we have over 170,000 specimens, in biobanks in Nigeria and also in Ghana. One of the things which our team also did was to develop a special neuroimaging software, which we used to archive our neuroimages. This is of relevant application, to ordinary clinical practice. And of course, neuroimaging data set are also an important component of a neuro biobank, especially in terms of doing precision stroke medicine or precision dementia care. Now, one of the things we did, and this is what every biobank should do, we looked at our practices in the Sarin Biobank and trying to align our practices with the IGC and ISBA best practices. And we were able to really certify that our biobanking practice actually met the international standard as set by the ISBA organization. And that's why this paper was accepted for publication. Now, we also have a fledged about the brain bank, which is coming up. Before the advent of this brain bank, there was no organized brain tissue repository in Southern Africa. So we have put Africa on the global map with this brain bank, and we have developed standard operating procedures, including how to code the brain and process the brain. And hopefully the activities of this brain bank, which were initially limited by funding, will pick up very shortly with some funding from one of our grants. And some of our trainees recently came back from the UK just over the weekend because they went for further training in Biobanking in Newcastle University. Then we have the African Biobank Patient Stroke Medicine LC project. This is a project that focuses on ethical, legal, and social issues related to neurobiobanking. Wanted to understudy what are the ethical, legal, social, anthropological, economic implications of the huge biobank that we're keeping, both in the Sarin study as well as in the brain bank projects. So through this study, we're able to identify and examine LC issues. We have developed interventions as well, and we have also built capacity, including training in genetic counseling and biobanking science. This was a workshop we had in 2021, two-day workshop that was very successful. We have also been able to identify challenges and opportunities in biobanking, ranging from using the socioeconomic, the socioecological framework, ranging from individuals to family, community, and societal barriers. For sociocultural challenges, some of the barriers that we noted were, barriers to acceptance of autopsy and postmortem tissue examination. Why do we found that in a study we carried out among people attending the geriatrics clinic, about 27% of them were willing to donate brains for research. And men and people with high education were more willing to donate brains for research. And the reason why people don't want to donate brain for research include fear of disfigurement, family does not agree, or the fact that they want their body intact for religious belief, so that when they come back again in reincarnation, none of their brain will not be missing. But facilitators include people knowing that brain research could save lives, and that body organs are no more needed after death. Now, some of the challenges again are infrastructural, funding, political will, having the, and of course, expertise, and all these challenges are being taken care of. There are also logistic challenges regarding biobanking, leading to evacuation of samples, sample storage space. Some subjects may refuse, there may be pressure on the field to package samples correctly, and then sample processing and barcode labeling. Each biobank will evolve solutions to address each of the challenges as they come up. Now, some of the interventions that were developed through the Biobanking project are involved using our community-based participatory research approach. We have established community advisory board in all our sites, and we go to the community to engage people about stroke, about genomics, and why they should take part in this research. We have also developed educational interventions, videos, comics, infographics, and case vignettes, all of which are being deployed to promote people to donate blood or brain towards brain banking. This is one of our community advisory board on our stroke project in Kano. They also oversee the biobanking program there. And then, of course, we engage with the community to educate them about stroke, about dementia, about risk factors, and how they can protect themselves, and also to come out and be evaluated for these conditions. So it's an integral aspect of all our projects. We have developed educational videos looking at different components relating to biobanking, neurobanking, stroke genomics, and also precision medicine. Time will permit me to show one of these videos, but they're all available on YouTube. So these videos effectively cover all the health issues, and they have been deployed for interventions in the community. We're building capacity, which is reported in this paper. We have also reported on the legal implications of these biobanking, including symbolic legislation that was uniquely described by our group. And of course, our ultimate goal is to impact the community, moving from nucleotides to the neighborhood. And we have also carried out a study in the community to find out how willing are people to undergo genomic testing for stroke. And people are really very willing, to undergo genetic testing for stroke. And people are looking forward to a time when stroke can be tested through become a point of care testing for diagnosis. Now, as I'm rounding up, I cannot but comment about the initiative of the College of Medicine in also developing a biobanking, which will incorporate all existing biobanks in a network, but also develop a dedicated space that I'll show shortly into a central biobank for the College of Medicine. We must give credit to the leadership and the pioneers of CREAM for having this original vision, which is still currently being pushed forward. Now, we're in the process of implementing this College of Medicine Biobank. We're following these 10 frameworks. We call them nuts and bolts, based on the ISBA criteria, step by step, to ensure that the College of Medicine Biobank takes off very nicely with full cooperation of all our PIs who have biobanks in their departments, in their units, so that they will join the ComUI virtual biobank network. Now, this is the new College of Medicine biomedical building. It's just opposite public health faculty. It's the new multidisciplinary biomedical research hub that houses the clinical biology laboratory, the Department of Medical Laboratory Science, and other proposed programs like the ComUI Multiomics Laboratory, the ComUI Bioinformatics Laboratory, and the ComUI Biobank at the basement. So this biomedical complex will synergize with existing ComUI research institutes like IAMRAT, IDI, ICH, and the Institute of Cardiovascular Disease, as well as with the Center for Genomic and Precision Medicine and other partners at home and abroad, so that we can actually domesticate and fast track and catalyze precision medicine in Ibadan as a leader in the sub-region. Biobanking is central to personalized medicine, as several authors have pinned in elegant articles, and it's been described as one of the new ideas that is changing the world. And I've shown this slide before, but I just wanted to show that before you can do your metabolomics, proteomics, transcriptomics, epigenomics, and genomics, you need specimen, you need DNA, you need blood fractions, you need tissue, you know, and this is why, and that means that's why you need a biobank to preserve the tissue. And I'm sure we all recall that DNA have been genotyped, even from Egyptian mummies. You know, these are bodies preserved for thousands of years. People are still able to carry research on them, you know, to raise new questions or use new approaches to answer old questions or answer new questions. So biobanking is key and central to precision medicine. And of course, data science at the other end. They're two Siamese twins that must always be together. I've shown this slide before, showing challenges, enablers, and benefits of precision medicine in Africa, and of course, to the West African sub-region. Now, I'll end this talk by reminding us of making mention of the Center for Genomic and Precision Medicine, which we started about 8 years ago. It's one of the offshoots of the East Africa program in Ibadan. And the component projects that are part of this center are all the studies that are currently doing one form of genomics or the other. They are all component members of this project. And I'm a deputy director after looking at research and training. And Dr. Adibi is also a deputy director, charge of governance. And then Professor Olabi is the director of that Gene Center. And is well represented online with 17 citations of PubMed. We also want to extend a hand of fellowship to our colleagues to join us to promote precision medicine in Ibadan and, of course, in the sub-region through the platform of the Center for Genomic and Precision Medicine. So, in conclusion, I have taken us through precision medicine as medicine of the future, and that future is now. Now, biobanking is an organized and systematic approach for storing biological materials for present and future mixed research, and there are existing and new biobanking initiatives in the sub-region and the COMUI, and that biobanking catalyzes precision medicine by making relevant biological samples available for multiomics, data generation, and of course, data science, serving to integrate all the multiple big data sets. So I thank you for your attention, and I want to acknowledge my mentors, teachers, collaborators, our various team members on our multiple projects, really the DSPs, Irene, Civ Genomics, LC, and of course our founders that are made it possible for us to do cutting-edge science, even in Ibadan. Thank you very much for your attention.
Thank you, Professor Akinyemi, for that thought-provoking and stimulating lecture. I want to thank you. I want to now to call on Dr. Bakare to handle the question and answer section of this seminar.
Thank you very much, sir.
Dr. Bakare, please.
Thank you very much, sir. Before we go into this session, we have to acknowledge the presence of many of our fathers in academics and research who actually grace this occasion. Let me mention some of them, but you are all actually appreciated. Professor Oguniyi, our former director, Professor Adimowo, former dean, Professor Olopade, vet, you are welcome. Professor Ariola, Professor Aneto Falusi, Professor Umigbodu, the former provost, Professor Walade, our former director, Professor Shinyowo, Dr. Joshua Akinyemi, Professor Arotiba, Professor Odole of Physiotherapy, Professor Fola Eson, Dr. Kotila, Dr. Akingbola, Professor Fentola, Professor Dr. Joshua Akinyemi, and so on and so forth. If your name is not mentioned, please, you are also welcome, please. Professor Akinyemi, you've done so well for us, and we're happy that we have somebody like you. Let me shoot the first bullet with a very simple way of answering this question to you, though so challenging. You've traveled wide all over the world, but your precision medicine and biobanking is actually restricted to West Africa, according to your title. From your experience, going from one country to the other, or comparing Europe, America, and other to West Africa, do you think Nigeria is there for now as regards the issue of precision medicine and biobanking, as more so data science?
Right. Thank you very much, Dr. Bakari, for that question. It is true that, I mean, not only myself, yourself, and all of us who have all been exposed to this cutting-edge science in other parts of the world, in the global north, and some in the global south. But you know, our passion is to change the narrative in our environment, starting from our institution to our sub-region. And that's why, you know, you and I have persevered with all the challenges. So our focus to change the narrative. Yes, it's a journey of 1,000 miles, and I believe we're very much on course. Time really does not permit me to tell the whole story of what we have achieved with the Siren project, but I can tell you that with Siren project, we're very much on course towards domesticating precision medicine in our soul region. Of course, I know that we're still challenged with some of the very basic issues, even about managing stroke in the hospital, the challenges of infrastructure, resources. Even when we're starting the stroke units, we have challenges of even being able to equip the place. So while we are not ignoring these local challenges to improve care for the ordinary stroke patient on our wards, we know that we must also not be left behind in the global journey towards precision medicine, which will ultimately be of benefit to the larger population. And one of the things precision medicine does actually is to detect disease, you know, before it develops clinical symptoms and signs, when the disease is still in intermediate phenotype stage. And this applies to virtually all conditions: diabetes, hypertension. If you pick people at that level through some biomarkers, maybe based on family history, based on some biomarkers, you can actually now introduce medications that can prevent them from coming down with a full-blown disease. And that's where we're going, and that will have greater population benefit. You know, and that's where precision medicine is taking. So precision medicine is taking us not just about treating, but prevention, better diagnostics, new therapeutics, and of course, improve quality of life for the average person on the street. Yes, thank you.
Thank you very much. Please, if you have any question or comments, you are free to indicate, or you put it down in form of a chat. We'll read it out for you. May I actually call on Professor Mayowa Owolabi? Sir, what actually will be the role of neurologists in this aspect of precision medicine and that of data science? Prof. Hello, sir.
Can you hear me?
We can hear you now, sir. Yes, sir.
Yes, but I didn't. Thanks a lot, but I didn't hear the question that you were trying to ask. Do you mind?
I'm actually trying to look at the role of neurologists in the area of data science and neurobanking.
Okay. You want us to comment generally on that?
Exactly, sir.
Okay. Thank you so very much. And congrats to Professor Akinyemi. Congrats to everybody who is part of the meeting, and of course to our mentors and teachers, particularly Professor Ogun, and to IAMRAT for hosting these monthly lectures, which has been very permanent and unveiling and very stimulating. Thank you so very much. So we're in the new era of brain health and what we termed the neurology revolution. The world has begun, I started realizing, that brain health is important for health, that without brain health, there is no health, and without health, there is no wealth, there is no development. So, of course, neurology is at the very center of this. And if we are going to leverage the knowledge and the technology and that humanity has achieved to promote brain health, certainly data science, multi-omics, and precision medicine have to be part of it. And what does data science do? Basically, is to collect information from multidimensional information from different sources, from individuals, and then bring it together and use the power of big data, so to speak, analytics. Use the power of big data analytics to convert this data into information that is useful. Information that is useful for prediction, information that is useful for identifying drug targets or novel interventions, information that is useful for prognostication. So, and because this information that you get from the data is based on where the data is coming from, that is why we must contribute data from the African continent. Otherwise, if the data is coming from Europe or from other places, it will not be adaptable or useful for us in Africa. So that is why we must, and in our own little way, with the Siren project and other projects, we've shown that we can collect data from Africa and come up with tools that can be used to predict some of the neurological conditions there and some of the solutions that can help reduce the body. So it's just an example which can be replicated across different fields in the College of Medicine and even elsewhere in Nigeria and Africa. So precision medicine has come to say is part of evidence-based medicine and is part of the new revolution. And another thing that is coming up is called synthetic biology, synthetic biology. And part of the drivers of synthetic biology include big data, includes that's data science, includes precision medicine, includes multi-omics. Now, synthetic biology is an immensely powerful tool that has more or less has infinite possibilities for good and infinite possibilities also for destructiveness. What it has done is that it has given us a double-edged sword, which we can use to almost cure any disease, because you can actually, with time, you'll be able to synthesize medications that can cure hypertension. Right now, for hypertension and for dyslipidemia, you can give a shot of a medication that can reduce lipid level for 6 months. So you only need to take a shot every 6 months and then you're fine. But I'm sure it's going to come to a point where using RNA technology, CRISPR technology, synthetic biology, you actually have a cure. We have a big body on the African continent. Hypertension, 77% of hypertension is controlled. 93% is not controlled. For so many reasons, it's not detected. People are not compliant and all of that. Even elsewhere, in developed countries, their control rate is also not fantastic. So with biologics, synthetic biology, so to speak, you can actually make organs, you can make new drugs, new molecules, but all of this will be based on data that is collected and utilized. And then it's also something that can open up our contribution and participation in the global knowledge economy. So it's really something that we have no choice than to be part of. So thank you for asking that brilliant question. We hope to stimulate other people to join us in the forefront of using biobanking, precision medicine, data science to advance the frontiers of knowledge and improve not just brain health, but overall health and well-being for our communities and society at large. Thank you very much.
Thank you, Prof. Professor Akinyemi, here is one chat. He said, thank you, Professor Akinyemi, for your very informative lecture. Drug abuse is on the increase in the country and is a great health challenge now in Nigeria. How will precision medicine contribute to the understanding, reducing, and treating this menace with the neurobank resource and intervention?
Well, thank you. Thank you very much, Dr. Bakari, for that very important question. I know there was an earlier program in the day, and there was a program earlier in the day on mental health, a conference. Apologies, I couldn't join because I was busy preparing for this talk. But I know that my brother, Mark Kunjela, spoke on substance abuse in that event. Now, I would like to approach the question from a socio-ecological framework. That when we talk about drug abuse, the problem is societal. You know, it's a societal problem. It's a family problem. It's an individual problem. Now, if you take the individual, there may also be a genetic predisposition to somebody, or epigenetics, for instance. We know that environmental exposures, things we're exposed to, have epigenetic effect, even on individuals and their offsprings. So the way precision medicine can contribute is, for instance, let me give this example. Children born to mothers who were exposed to the genocide in Rwanda, it was found that there was a higher prevalence of post-traumatic stress disorder in these children. There was a study that found that, and the key was epigenetics. There was some evidence of hypomethylation of some, or some regions of the genome. So how can precision medicine help? As we are managing these individuals, the young men and women who are abusing drugs, we can take blood samples from them. You can store their blood, store their DNA, store their plasma, and run markers. Look for some markers. There may be genetic markers, there may be familial markers in them that make them to be prone to these drug abuse. In addition to all the other interventions, clinical behavioral therapy, and of course counseling and all that. So there may be a biological component which may help us to even protect the next generation. So that is how I think precision medicine, and may also help us to understand why there are differences in the risk or tendencies, maybe between comparing indigenous Africans to other Asian population. I mean, it's a global problem, really. So is it a problem of the African ancestry individuals? We know it's a problem of Blacks in the US. Why is it also a problem, is a problem in Africa? Is there some genetic predisposition apart from social determinants of disease? So there's always a way that, so the biobanking would help us to explore the biological dimension to this menace. Thank you.
Thank you very much, sir. The message here is from Olabode Mutiloye. Say wonderful delivery, Professor Akinyemi. Just actually appreciating you. Another person said, that is Samuel Olawale. Said, thanks, Prof, for this great opportunity. And many more. So if you have any question, please, you can indicate or put it down in form of chat so that we can put it across to our guest lecturer, Professor Akinyemi.
Yeah, Dr. Bakari, we have people's hand up. Can you call those who have their hands raised, please?
Okay, let me see.
There's Akinyemi Ojeshino, Ifeoluwa, and our Professor Falusi.
Let me see.
Check the participants' column.
The participants, okay. Okay. Professor Falusi, ma, please. The floor is open to you, ma. My former director, Dr. Akinyemi Falusi, please. Hello, ma.
Maybe you can call Ojeshino before she joins.
Okay.
Hello. Back.
I'm here.
Okay, ma. Yes.
Did you
Okay,
want me to? I didn't know you wanted me to speak. Thank you very much. I just have one or two comments. The first comment to make is that I'm so appreciative of the effort of the team that is doing this work. This is something we have looked forward to for many, many years, those of us who started many years ago. And I'm happy that we are getting to this point that we are. So Dr. Mayowa, Professor Mayowa, Professor Akinyemi, I'm clapping for you right here. That's the first thing. The second point I want to make is that you have actually answered two of my questions, because in 1983, when I was doing my work, we could not answer the question of who owns the data, who owns the sample. We could not answer. We took our samples from here, worked on it, took it to UK, US, and so on, and did the work. And most of the samples were biobanked in the other foreign countries. But at that time, this issue was not up. So you just did your work very well on the bench and got your data, and you were happy coming back. And subsequently, you did some publications with them, but the data resided over there. So we are thanking you now that the dynamics is changing. So we can leave our biobanking on this side and go to do some work with them over there. So that's one question answered. The second part I want to talk about is the fact that you are seeing the differences in the genetic admixture that happens throughout the world. In the 80s, when we did the work on sickle cell disease, Professor Esson is here on this platform. We saw the differences that were occurring between the Black Americans, the East Saudi Arabians, even within East and West of Saudi Arabia. There was a difference in the genetic structure of sickle cell disease. The same thing is what we saw here in Nigeria, that when you look at India and you look at Nigeria, there is a world of difference between the structure and the haplotype stratified profile. So it is good for us to concentrate on what is really happening in the African diaspora, in the African setting. And I'm happy that you are doing this work, because even within the African setting, we are going to see a lot of things happening in differences between the West African sub-region and the East African sub-region. So there is more to come from this. If you look at the breast cancer that is happening in genetic research unit, Professor Lokpade is in charge of that. We can see the early onset in the Nigerians, in the Africans, compared with the Black Americans and the Caucasians. There is a difference between the three groups of what we are seeing there. So this genetic diversity is actually a good thing that we can now pinpoint in our own area here. What can we contribute? Can we bring out the facts here, which is what you are trying to do? So we are very happy that you are doing this work, and many more will join you to bring out the African scenario and compare it with, as those are bringing theirs from over there, we can be bringing the African sub-region. The only question I then want to ask you, those are comments, is that with this biobanking, I know it's on stroke and you are doing a lot of good work. Is it possible that those of us not doing work on stroke can join you and have a biobanking that has different sectors, like sectors on sickle cell disease, that we can put a biobanking sector. Thalassemia, we can put a biobanking sector, so that within this enclave of what you are trying to bring out, which is going to be a beautiful thing, can we have this along with you so that we can work together and we can have this African science that will be well comparable with other in the other social areas of the world. Thank you very much. That's my question.
Well, thank you. Thank you so much, Ma, for your very elegant and inspiring comments. And I must commend you, Ma, that even in retirement, you remain a source of inspiration for us. And I want to assure you that by the grace of God, the labors of our heroes, past and present, your labors shall not be in vain. God helping us. Now, to answer your question, the slide I'm showing is the answer to your question, Ma. It is true that the current biobanks that we're running as a group, we have banked materials, DNA, red cell concentrate, plasma serum, primarily from stroke patients. And now, more recently, we also started collecting samples from patients with dementia. And Ibadan is collecting samples from non-African countries. So it's a big project that we're also running. The largest study of dementia is started and is running. But beyond that, even I must say that we have even supported colleagues. People have stored things, in our freezers based on understanding. So we're very open to supporting colleagues. But beyond that, the College of Medicine Biobank, which is being championed by our provost, and of course, originally initiated by Crane, but now being championed by the provost, I chair the College of Medicine Biobanking Committee. It's a committee of 10 very hardworking and passionate people. In the basement of this building that you are seeing on your screen, there's a space that has been dedicated. And we're already in partnership with some persons abroad to support us. And we already have some funding. We have already contacted IAMRAT. They are coming to visit this biobank as the first step towards establishing it. So this biobank will be open to all researchers in the College of Medicine. If you have your freezer and your alternate power supply, there's space for you. If you don't have a freezer or alternate source of energy, you can also rent a space. It will be a cost that is affordable. And in fact, we're encouraging researchers that when you're writing your grant proposal, please put budget for biobanking in your proposal, so that when that money comes, it's deducted from source, and that's how we can grow biobanking in the College of Medicine. That's a big vision. That's the big picture that we have. So hopefully, and of course, the other thing we're trying to do is that we're trying to network all existing biobanks in biology, in pathology, microbiology, IDI.
Hello?
Maybe you should move.
Professor
Okay. Maybe you should look. Okay. Can move away from that place. Thank you. Can we call on?
May I suggest that we please make our questions short or brief, and also the respondents, how do I put it? Our lecturer, you make the comments also short so that many people can be attended to, and also time. So we don't want an empty house later. Thank you.
Okay, ma. So now let's go to Dr. Akinyemi Ojeshino or Professor Akinyemi Ojeshino. Please, over to you.
Good afternoon. Thank you, Professor Akinyemi, for your wonderful presentation. I really enjoyed it. I came in a little bit late, but I'm looking forward to listening to the recording of the beginning. So the question I have, I will admit, may come from a standpoint of relative ignorance. So really, I'm looking for you to help me dispel that ignorance, if that's what it is. I noticed that your wonderful work on polymorphisms and stroke have been focused largely, and by the way, not just yours, but similarly many works actually have been focused largely on ischemic stroke rather than hemorrhagic stroke, considering the fact that the incidence of hemorrhagic stroke is actually higher in Nigeria and many parts of the world too. I wonder, is there something particularly peculiar about being able to study ischemic stroke more than hemorrhagic stroke, or is it just opportunity or just resources or collaboration? It's just really curious to me that, and once again, this may be ignorance, and that there's much more work than I know, obviously, but it's just curious, and I wanted to get your comment on that. Thank you.
Hello, Professor Akinyemi. Hello, are you there?
Oh, yes. Oh, okay, thank you very much. Yes.
Do you suggest that we take all the questions so
okay,
that
yes, that's fine. Right, that's
thank
okay.
you very much.
Let's afford the questions so I can respond to you.
So just that. Thank you very much. We go now to Dr. Ife. Hello, Ife.
Yes, sir. I'm here, sir. Can you hear me, sir?
We can hear you now.
Thank you so much, sir.
From biochemistry.
Yes, thank you so much for the lecture, Professor Akinyemi. I would say it's eye-opening for me, especially with respect to novel African ancestry locus that you presented from other studies and whatever that you also identified. I'm just curious, and I want to ask a follow-up question. Maybe it's in your platform or in your future agenda to actually connect biobanking, tissue biobanking, not actually the blood, I mean the brain tissue, to translational science. I mean, relating to working with basic scientists, because I know whatever you find out from your genomics, for instance, you are talking about ApoL1, one needs to know what is the function, like what do they do, how do they circumvent, how do they become protective. These are mechanisms that cannot be explained that cannot be explained fully by genomics, but basic science has to come to the rescue. Do you have future platforms to actually collaborate or to actually have a platform that basic scientists can come in and make a request for samples to actually compare what they have in humans, maybe with what they have seen in rat or mouse or Drosophila model, so that they can actually bring this approach that you have domiciled in Africa further. Because nobody will come from the US or something to actually do it for us. We have to devise an approach. Thank you so much.
Thank you, Ife. So we go to Professor Dr. Adekunmi Odekunle. Over to you.
Thank you, moderator. Once again, lending my voice, I want to thank Professor Akinyemi for the well-articulated lecture. Mine is a comment and a question. First, Professor Akinyemi has given us a gold mine of data that I think will raise some questions and opportunities for cross-disciplinary approach, which I think should start from the College of Medicine. And what do I mean by that? Besides the biobank, the information as presented is a large data set that I think that can be useful for a lot of researchers. And we complain so much about data in Africa. We complain so much about funding to recruit respondents. But here is data, and I think that this is an opportunity for researchers. Being familiar with most of the data I presented, there are several multiple information ranging from multi-omics, the lifestyle factors, social demographic information that can be useful, for even, for PhD students for their masters and thesis, something and things like that. And just to ask you, sir, two things. One, you didn't talk about data access framework for this, for some of this repository you have. I think that the data access framework is necessary. Why? Because you will also be very much aware that researchers even from the global north are currently interested. They are very much interested in this data set and information we have presented, where they are making cross multi-accessory comparison, African data set, and things like that. And secondly, I think also that you didn't mention also about. I'm sorry, maybe I missed that. How we can also use this genetic information that we have in Mendelian randomizations to improve our risk factor prediction models and design suitable interventions for primordial prevention in some cases improve prognosis of patients. Thank you.
Right. Professor Akinyemi, before you go on, one question too from the chat from Dr. Adewoyi. Huge appreciation for the wonderful presentation, Professor. Could precision medicine help in reducing the misbehavior leading to the menace of rape, especially raping of minors and subsequent dismemberment of their victims in the name of ritualistic mentality? Did you get the question? Hello, Prof. Did you get the question? Hello, Prof.
Professor Akinyemi. Professor Akinyemi.
Well,
likely to be internet connection. Okay, Prof. Did you get the question I read last?
It's apologies. The network challenge.
Yeah.
So I'll be
Sorry.
Yeah, to know more about the family history of such a person. There's a familiar history of such and social behavior. It may be, there may be genetic or epigenetic components why the person behaves that way. So there may be then be a biological explanation. But I'm very sure that 90% of the risk of that kind of behavior will be largely environmental, lifestyle, upbringing, or sometimes even determinants that are set early in life. We're not understanding that many diseases actually have their foundation in utero or early life. Changes in morphology of the brain networks, brain neuronal migration, how brain networks are formed. Many of them, they determine how people develop behavioral disorders later in life. So there's more that means there to that question. Right. So there may be biology component, but of course we need materials to understand that better. Now, Dr. Kikumi, thank you very much for your elegant comment and question. You'd agree with me that you can't always cover the whole range of topics in a 1 hour talk. Yes, indeed, the H-Chemical Consortium has set in place material and data transfer agreements, including data sharing agreements, primarily to protect African researchers. So they were not bulldozed into giving up our data with colleagues from the global north. So we follow, I must tell you that we've been under tremendous pressure, sharing study in the last 5 years, people wanting our data. We have been protected by the East Africa guidance that we cannot share our data until we have published our GWAS study, which is now almost to be out, should be out in print, hopefully before Christmas. So there are frameworks to protect you from sharing either your genomic data or your phenomic data. And of course, the whole remit of data science is also about that, to protect you so that you're not, it does not become another round of slavery. Your data going out and then being used to discover drugs that they come back to sell to you. So there are frameworks to protect data sharing or material transfer now. Now to my very good friend and brother Akinteshino's question. Yes, ischemic stroke is not the only subtype we're studying. We're also studying hemorrhagic stroke, but our first GWAS has been on ischemic stroke. Even the candidate gene study have been on ischemic stroke. We are now shifting attention to also examine hemorrhagic stroke genetically, given the incidence of small vessel disease in the environment in particular. We think that there may be genetic variants that also predispose to the higher prevalence of hemorrhagic stroke in the environment. So the next 1 or 2 years, there will be outputs on hemorrhagic stroke from our team. And then I think I've dealt with all the questions. Now, about tissue availability for further study, you're right. You can't answer all the questions from blood fractions alone. You need tissue. For example, if you're doing epigenomics, what you get in blood may not be exactly what you get from brain tissue. I very much agree. That's why we're also making an effort to store brain tissue, fresh frozen or paraffinized blocks. These are all gold, golden materials to answer new questions or old questions in new ways. We can run microarray, we can run transcriptomics even on brain slices. We can run single cell. We can run single cell assays. Transcriptomics, even that will give you myriads of molecules that can now use to determine new biomarkers or new treatment. So there's a lot of potentials. And of course, we're very willing to collaborate. And of course, we have a multidisciplinary team. On our team, we have basic scientists. We even have social scientists on our team, ethics experts, health education experts. So our is a multidisciplinary team that has enriched our productivity in the last decade. Thank you very much.
Thank you, Professor Akinyemi. We are sorry, a lot of questions are there, but you can look at the time, almost 2 hours of the lecture. Please, your questions are actually noted. Just brief information to all that if you are interested in handling CME credits, you can please indicate on this platform. My directors,
yes.
Over to you, ma. Yes.
There is the register, which I've sent the link. We'll even appreciate, even if you don't need a CME, to also register. It will help us with our record. And also if we need to get in touch with you for subsequent activities of IAMRAT. But Dr. Bakare, please, can you, I have two questions here. I think they're interesting. One from Jomaka and Olawale. I think we can then round up with those two, addressing those questions.
Okay. Let me just read the questions out, ma, so that we can, about 3 or 4 questions there, yes. About 3 or 4 questions. Yes, ma. So thank you, Professor Akinyemi. To what extent is secondary data from this project available to African researchers, especially early researchers? That is one. Another person came in with 2 questions. He said, thank you, Professor Akinyemi. In general, under ideal conditions, how long would the bio specimen last in a biobank? Could be bio specimen be stored indefinitely, or is asking whether it could be actually stored. Then the other question he has is that could you provide insights into the specific storage conditions implemented to ensure the long-term viability of bio specimens in your biobank? I think these are the questions. Then the other one will say, we are watching from UK. Please, we need the CME too. Brilliant presentation, Professor Rufus Akinyemi. That's from Olayiwola Ajileye. So I think that's why I have not asked too much on the role of precision. Okay. With our time, anyway, can you just say 1 or 2 things, Prof?
All right. Yeah. Let me thank the president of our ECOMA UK branch, Dr. Olayiwola Ajileye. Thank you so much, sir, for joining the call. You were asking about role of precision medicine in mental health disorder. You see, I have decided to use stroke and dementia as a platform to discuss that topic because those are because of limit of time, one, then those are our areas of focus where we have some data from this environment. Certainly, precision medicine is applicable to every field of endeavor across different specialties. So I think I'll just stop at that. Now, about storage facility or storage conditions for bio samples. Well, those are very technical questions, which I don't think I can speak to in this forum. But if you want to do biobanking, get in touch with relevant teams, then we can share our protocols with you and share our experiences with you. Right. Then about secondary availability of data. We have many early career researchers who are working with us on our team. So if you're interested in growing with us, as we're growing, we want to carry others along, please get in touch. We're happy to, we have data and manuscripts discussions where we all learn and we grow. So if you're interested in growing, working on our data sets, so I think you can reach me or reach Professor Wolabi. Very happy to, we're very keen to grow the next generation. So you get in touch with us. I think I've addressed most of the questions.
Yeah.
Yes, sir.
I think you've done justice to virtually all. Thank you very much, Prof.
All right,
my
sir.
director, ma. Hello, ma, my director.
Just a minute. I just want to put the link again for registration. I see people putting in their emails and so on. They should please access this link. I've copied it again. And there you can put all the information. Yeah, I think it's been a very long but very worth while time. And want to thank Professor Rufus Akinyemi. I know there is a lot that you would love to have shared with us about biobanking and precision medicine. I'm sure we can have another, how do I put it, another edition, let me put it that way, where you come out with results that I think would again help people in this balance of research. I think the vote of thanks has been done partially, and since I've been given this time to talk, I want to appreciate everybody that have joined this lecture series. A lot have been said about our former director, our former provost. We also have our legend, like Professor Esson, and others who have joined this lecture. I think we are we should really appreciate you. It's not a convenient time for some people, even those in diaspora, but they have actually created the time and they've been with us all through. I want to appreciate our provost, who is ever supportive. She's very particular and proud, I would say, of our lecture series that has been going on consistently over the years. And she really holds IAMRAT in high esteem. I also want to thank those who have joined from outside the country first, our African scientists in diaspora. We have people from the UK, those into drug development, those into biobanking. We have people from South Africa. We have those from Kuwait. We have people from Canada. We have those from US, France, and maybe the one from France. I must say that he's one of the beneficiary of the data from the siren. He's going to do his PhD from the data that has been mentioned. I would not want to take much of our time mentioning names anymore. So please just know that you have been highly appreciated for being part of this. I want to also mention that in terms of biobanking, there are a lot of ethical issues that we should always bear in mind, just as been said. And the issue of brain bank, it's unfortunate Professor Rufus cannot go into it. It's an interesting area that I'm sure many people would have loved to hear in details, but we'll look for a way of bringing it back and look at the social aspects of biobanking. or maybe socio-legal aspects of biobanking in one of our lectures next year by God's grace. I want to mention that this November lecture would be the last for this year. Usually, you know, in December, people close early. And also we need that time to sort of write our report, assess or evaluate our performance, and plan ahead to improve on our activities in IAMRAT. So I want to thank everybody for being part of our success in IAMRAT this year. And we hope that, please don't forget, by January we are back. And we also extend our invitation to those who have interesting research findings or research activities that they think people would like to hear, to extend that to us. We are open to support people to disseminate their research findings, and also with special focus on even translational research. But lastly, I want to really thank Professor Rufus Akinyemi and his team. I'm happy Professor Wolabi also chipped in. I know they are working together. Well, Professor Akinyemi, I think you have really done a great job at delivering this lecture, sharing very important message to researchers and even lay people, because I know I can see lay persons also joining. And you also tried to deliver the lecture in such a way that people who are not so knowledgeable about genetics and so on could really understand. That is very important in any public lecture, and you did that so well. And also I join others that have commended all your activities and also this lecture. I think you have really done us proud. IAMRAT is proud of you. College of Medicine is proud of you. And UI too is proud of you and what you and your team are really doing. You are really in the forefront of biobanking, as you have really demonstrated today. And I hope that IAMRAT would also be in the forefront of the biobanking, college biobanking initiative, as you have mentioned. So on this note, I say thank you very much. And we hope to see January ending as usual. And but however, in between, should you have anything to share with us, you can just write the director, or we have our website, and you will see email addresses of people you can contact for anything related to IAMRAT. So thank you very much, and I wish you a wonderful evening.
Thank you very much. Thank you. Thank you.